Hello, good afternoon, everyone. And welcome to Coffee Science Seminar series today. Olumide. I am Femi Akinsami. I'm based here at the University of Queensland, Center for Horticulture, Raw Science here in Coffee. I'm located in ASP Ecosciences Precinct here in Burger Road. I primarily work on macadamia, diseases of macadamia as a plant pathologist, and I've had to work on various aspects of pathogens, including the soil-borne pathogens and flower blights or area-borne pathogens in macadamia. I've also worked on various other cropping systems, but in most we have a seminar that's also on macadamia. Hence, I'm sharing this seminar. Acknowledgement of country. The University of Queensland acknowledges the traditional owners and their custodians of the land on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australia and global society. Before we start, I'd like to go through some housekeeping rules. So this seminar will finish at 1 o'clock, and there will be time for question and answer at the end of the presentation by our presenter today. However, I would like you to please write your questions in the Q&A section at the tab below your screen. And at the end of the presentation, I will ask those questions on your behalf. Today, we have a postdoctoral research fellow, Olumide Jeff-Ego, who is presenting part of our work she's been doing in macadamia. Olumide has been a research assistant. Was a research assistant with me for a few years before she started her PhD, working on Phytophthora in macadamias, and she's received certain awards, including being named the Woman of the Year by the Australian Macadamia Society in 2020. Our research during her PhD has related to a lot of new things, new frontiers, and she was fortunate to receive the Vice Chancellor, University of Queensland stimuli package to retain new PhD students or newly completed PhD students. So she's back to expand on what she started doing during her PhD. And today she's going to present part of her work on Phytophthora in macadamia, a growing trait. So without further ado, we'll ask Olumide to present. We can't see you and your video is off.
Yes, I'm actually wondering what's going on. It says my host has stopped my video.
No, I didn't. Okay. I can hear you though. So.
I'm trying to start my video and see. Well, it still says you cannot start your video because the host has stopped it.
Okay, just continue to present.
Okay. All right. Sorry that you can't see my beautiful face today. And my name is Olumide Jeff-Ego, and I'm a postdoctoral research fellow for the Center for Horticultural Science, Coffee. And I'm excited to present today on my research And before I proceed, I'd like to acknowledge the traditional owners of this land and their custodianship of the land on which we meet. And I pay my respects to their ancestors and their descendants, past and present, who continue cultural and spiritual connections to the country. And we recognize their valuable contributions to Australian and global society. Yes, so today I'll be presenting on Phytophthora, my favorite pathogen in my favorite crop. And I've titled today's presentation as Phytophthora in Macadamia, a growing threat. My presentation today will run from the background of my research, the crop and the pathogen, down to my research and my research outcomes. And I would share with you the significance of my findings and a few conclusions. Yeah, so just a quick background of the crop macadamia. The crop macadamia is native Australian commercial crop cultivated for food, and it's cultivated for their edible kernels. There are 4 species of macadamia, and these 4 species are native to Australia and along the central coast of the southeastern Queensland and northeastern New South Wales. And they occur naturally in the wild and with their natural hybrids. Just, okay, lovely, thank you for doing that. All right, so I'm going to my next slide. So just a quick overview of the crop macadamia. It says here, I have the 4 macadamia, there are 4 macadamia species like I earlier mentioned, and 2 of these species have been developed and cultivated commercially for food because of their yummy edible kernels. However, 2 of the other 4 species, that's Macadamia ternifolia and Macadamia jansenii, are not cultivated commercially due to their high cyanogenic content, and they're not actually edible. So you must see a lot of commercial products, including macadamia as additives in confectionaries, in lots of cosmetics, and a wide range of products. And that makes macadamia really, really popular. And I'm sure you love yummy macadamias when you taste them. Well, in Australia, commercial production ranges from Mackay down to Nambucca Heads, and the largest production region is in Bundaberg. But there are offsprings of lots of other production regions in Margaret Rivers, that's in Western Australia, and some pop-up places, production regions in Tasmania and Adelaide as well. Just a snapshot of the Australian macadamia industry. Well, Australia is one of the world's largest macadamia producer, alongside South Africa, which is currently the leading producer of macadamia. We have about 800 growers covering over 25,000 hectares of land. And all of these productions at the farm gate is valued at about $285 million. Other production regions in the world, because macadamia production is spreading, and macadamia can be produced in frost-free tropical and subtropical regions of the world, including in places like Guatemala, places in Africa, South America, and in some other places in Asia. There are several diseases of macadamias, but the major diseases that is significant to production is Phytophthora diseases and husk spot. And today I will be sharing with you on Phytophthora disease in macadamias. And you know, Phytophthora diseases have been reported in most macadamia production regions all around the world. So that leads me to just an overview of what Phytophthora has been doing in macadamia and where it has been over the time and where the report has been. Here I have about 5 Phytophthora species. which is Phytophthora capsicum, down to Phytophthora cinnamomi on the slide. And in different parts of the world, there's been reports mostly in Hawaii and in Australia. But I would like to point out that Phytophthora cinnamomi is the most reported, and which caused stem canker, root rot, branch dieback, and slow decline. Just a quick overview on the pathogen again. One of the species of Phytophthora that causes diseases to macadamia is P. tropicalis, Phytophthora tropicalis. And you would see on the photos here that some guys are actually wondering what sort of devastation happened to the tree. And here you have the tree dying, showing gummosis. There is dead, brown, waxy, defoliated, well-retained leaves on the tree. And well, the final stage of what you see here is tree death. And that occurs within 2 to 3 months post-infection. Again, Phytophthora EVA, which causes macadamia quick decline, also reported in Hawaii recently in 2020. That's to show that there is more incidence of Phytophthora species that can cause diseases to macadamia. So the symptoms include abundant dull, light green to dark brown leaves. You have the browning of the petioles along the advancing leaf margin, and then you have some defoliation. The difference between these caused by Phytophthora EVA and Phytophthora tropicalis is that there is some level of defoliation which precedes death, whereas in Phytophthora tropicalis, most of the brown leaves are retained on the tree. Okay, so we have another one, which is the most popular and the infamous one, which is Phytophthora cinnamomi. That causes slow decline and reported mostly in most of the major producing regions, which causes root rot, and the symptoms significantly shows as branch dieback, chlorosis, and sparse canopy. And there's significant loss in the yield, the capacity for the tree to actually produce macadamia nuts as well. And then how does this happen? You have spores of the pathogen, Phytophthora cinnamomi, splashed onto the trunk of the plant, or probably infecting the roots, which causes stem canker. And you see it as gummosis shown in the picture from left to right here on the slides. Well, all of these diseases have a management to it, and currently management practices include disease chemical application and cultural practices, including chemical applications include the trunk injection using systemic fungicides, and sometimes you have copper protectants just to cover the back of the infected plants, so that to alleviate the infection progression. And then you have cultural farm good practices such as good drainage and using of disease-free planting materials. So it's essential to know what the economic impact of these organisms, these pathogens had to macadamia production. In Australia, it has been quantified that about 20 million loss in productivity is potentially going to happen if there is Phytophthora infection across the major producing regions in Australia. And then there's lots of reports of thousands and hundreds of mature trees and poor establishment of the trees. So the presence and the impact of other Phytophthora species are actually unknown. I have mentioned about 5 that I know of and that has been reported. The most reported of them is Phytophthora cinnamomi, as I mentioned. And this leads to me questioning what is out there, what other Phytophthora species is out there, and that leads me to what I'm going to show you now that the number of Phytophthora species worldwide is increasing. And that's because, well, we have more availability of technology, DNA sequencing technology. And now we have over 187 species of Phytophthora that are possibly causing devastation in lots of plants, and over 120 of them are known as plant pathogens that are already confirmed. And the trend is projected to be increasing. So the question is, are we really at risk? With international trade and global transportation, we know that there would be intensification of these aggressive new and exotic Phytophthora species that might possibly cause diseases to macadamia and probably other crops. But in the context of this presentation, we have, there has been a test done in vivo in the US by one of the scientists, and it's been tested that Phytophthora ramomi is may potentially cause devastation to macadamia. And I must mention that Phytophthora ramorum is actually a pathogen of significant biosecurity risk to Australia, as well as Phytophthora tropicalis, which has been reported in Hawaii and in China. Again, the question keeps bothering me that, are we at risk? Are there many more Phytophthora species in the commercial orchards that we do not know of that are causing significant devastation in the orchards, and we're just only implicating only Phytophthora cinnamomi? And are these Phytophthora species, if they are present, are they pathogenic to macadamia? And so the question is, can we identify also the Phytophthora disease-resistant macadamias if these species of Phytophthora are able to infect macadamia? There is a constraint to that, and that's because of the longevity of macadamia breeding cycle. And so it's very, very difficult to be able to test out these pathogens on what they can do to macadamia and how much impact they can have to macadamia. So I had on my mind when I started this research to get answers to the questions, and I hoped for the outcomes, such as to determine the prevalence and identity of and pathogenicity of Phytophthora species associated with macadamia, especially in Australia, and to also see how I can shorten the length of the breeding cycle, the impediments, for testing out all the macadamia materials that I'll be including in my experiments. And I should also test the reaction of open pollinated macadamia cultivars using implanter stem and root assays, which I would be able to compare to be similar to in vivo assay that I would develop. And also test the wild genotype of macadamia species and their natural hybrids as source of resistance to Phytophthora species that I evaluate. So my first goal was to determine the prevalence and pathogenicity of Phytophthora in macadamia in Australia. And I'm excited to let you know that I have published one of the articles in Plant Pathology, which is the prevalence of Phytophthora species in macadamia orchards in Australia and their ability to cause stem canker. So in doing this, I surveyed the commercial orchards in Australia across Queensland and New South Wales majorly. And I collected samples from August in 2016 down to December 2018. And I collected field data, including data such as the cropping history of the farm, the tree health status, and some symptoms of Phytophthora diseases and what I observed on the farm. So I collected tissue from the tissue samples. I collected from symptomatic samples and asymptomatic samples For symptomatic samples, I collected 45 tissue samples, and from asymptomatic, I collected 10. And from the rhizosphere, you know, in the pathogens, they live in the soil. So I collected the soil samples from symptomatic, around asymptomatic tree, symptomatic trees, and other possible sources of inoculum. That is some drainages leading into the orchards, some nursery samples, plants, some from runoff, and some from other vegetations and other crops cultivated nearby the farm, the macadamia orchard. So on the, this formed one of my research chapters actually, and I've also published it. And from my collections of the rhizosphere sample from Queensland and New South Wales, I had about almost equal samples, 182 in Queensland and New South Wales that I collected. And in order to determine the presence of the pathogen Phytophthora, I've used to be able to get them out of the soil samples, I've used some baiting techniques, which I use some plants which are known susceptible hosts, including umbrella plants, Geraldine wax, and New Zealand blue lupins. And I've isolated them on V8 juice agar, amended with antibiotics. And using DNA sequencing together with, combined with morphological features, I also identified these organisms that I isolated from the baits. So I also confirmed the identity of the organisms using identify tools, which is a database for Phytophthora. So again, I determined the mating types of all of these isolates that I collected. A couple of the isolates I collected using the testers of known mating types, which I collected from the UQ, which I obtained from the UQ Phytophthora collection. And I paired them together so that they produce the sexual spores, which is useful. And so from my collection, I identified a whopping 8 Phytophthora species. And of them, I have Phytophthora multivora, Tetraphylla, Nicotiana, Cinnamomai, Sojae, and down to one of the Phytophthora species that I have not identified down to species level. So based on my results, I identified the mating types of some of the isolates I collected, and I saw that of the mating types, the most prevalent was the A2 mating type, and we had a few A1 mating types. And in some few instances, I had some of these, in some of the locations, I had both mating types existing, including places like Palmer's Channel, Maritri Door, Woolumba, and in Gympie. For other of the heterothallic species of Phytophthora that I collected, which is Phytophthora nicotiana, Pseudocryptogia, and Tetraphylla, I have only A1 mating types in my collection. And this is very, very important for the abundance and the distribution and population explosion in case of an epidemic. And is also, this information is important for control as well. So from the macadamia tissue samples, I collected mostly Phytophthora cinnamomai again, which constitutes about 82% from the symptomatic tissues. And again, I collected Phytophthora multivora from stem canker as well. This table here shows that most of my collections were of Phytophthora cinnamomai, and there was significant collection of Phytophthora multivora. And so all all my collections were both from symptomatic, asymptomatic, and other possible sources of inoculum, which shows that it's possible that the nearby vegetation might be a source of inoculum, a pathway for that causes the presence of the pathogen Phytophthora into the orchards. So from my results as well, in terms of the species distribution by geographical area, I have the most frequently and the most widely distributed from the table below here is, it shows that the most widely distributed is Phytophthora cinnamomi, and the highest diversity, and it shows that it has the highest diversity towards the north. That might be maybe a collection bias, but that is what I have. That was the result I got from my collections. And so I have Phytophthora multivora also clustering towards the southern part, which was most collections that I collected from New South Wales. So in order to test for the pathogenicity of the Phytophthora species I collected, I have used an in vivo detached macadamia leaf to run the test on a non-susceptible cultivar to confirm the cost postulate. And also I tried my hands on the implanter stem inoculation, and I measured the lesion length. So in terms of the pathogenicity trial, I observed that the most significant disease progression that I observed was from Phytophthora multivora and Phytophthora cinnamomi. And here I included palmivora as the control. So it shows that, which has been said to be a leaf, macadamia leaf pathogen. So in my in vivo implanter inoculation, again, it shows that Phytophthora cinnamomi and Phytophthora multivora caused significant amount of disease. So here again, this just to demonstrate the significant progression of the stem lesion based on the photograph you saw previously. So some conclusions here. Here, from my study and from my findings, I found that Phytophthora cinnamomi and multivora causes diseases in macadamia in Australia, and I have established that. And here, it is the first report of Phytophthora multivora to cause diseases in macadamia, to cause stem canker. And also, I observed there were 2 mating types in the same orchard, which is risky to effective control of if there is a population explosion. So, in order to do to make sure that I'm able to screen large germplasm, I tried to develop a rapid assay to assess a large number of, to determine the varietal susceptibility of major macadamia commercial cultivars and to Phytophthora species. And in order to do that, I used a detached plant part, which I mentioned earlier that I used the macadamia leaves. But before I arrived at using a macadamia leaf, I actually tested other methods to see if it works and if it was suitable to be able to rapidly screen large amount of germplasm. And so I hypothesized that detached plant parts is a suitable tool for rapid screening of varietal susceptibility to Phytophthora in macadamia. So in order to do this, I actually made sure to test for suitability in terms of, is the assay high throughput? Is it simple? Is it fast? Is it inexpensive? And is it repeatable? And is it fit for use? So I tested on did a lot of inoculation techniques, a few of them wounded and non-wounded leaf assay. And then I did detached twig assay as well. So from my results, I saw that the wounded and the non-wounded leaf assay would not work. And I also did and it's not consistency, and it's not consistency for disease severity. For the one that I did, it is called that worked and was repeatable, it was simple, it was fast, and it was fit for use, was the detached leaf assay. I've called it whole leaf deep assay, which based on my results, the symptoms was visible at 7 days post-inoculation, and it showed a distinctive progressive symptoms development, which you can see as which starts as golden brown to soft dark brown spot that progress to on the leaf blade. So in order to optimize my assay, I chose about 4 cultivars to test, and on the wild macadamia plants, Macadamia, integrifolia, and tetraphylla. And then I selected based on the leaf age, the amount of inoculum, and the time it takes to take the data. And from my results, I observed that there was no significant difference between the use of middle-aged macadamia leaf and the young succulent leaf. So I tested for the influence of the inoculum concentration as well. And based on my results, I observed that there was no significant difference between using 3 to 5 mycelia agar plug, but which could influence the result that I would get. So I compared, based on my experiment, I compared the in vivo assay against the field varietal susceptibility and based on that, it had similar outcomes based on the pathogenicity on the 4 cultivars that I used, commercial cultivars that I used, is similar to the field disease expression as collected by Akinsumi et al., and which you can see based on the charts here. So some conclusions here is that I have found a new strategy to identify genetic resources against Phytophthora developed. And I have also gotten a good assay that's proven to be simple, high throughput, and effective. And the new assay supports the field performance of commercial cultivars based on what I have done in the lab as well. So another part of my research was focused on the in vivo and implanter assay, which revealed that the progenies of macadamia cultivars, that there is a similarity in the heritability. So here I hypothesized that the disease severity level of plants from open pollinated seeds are similar to clonal materials. And so I determined, my specific aim here was to determine if the reaction of open pollinated cultivars using implanter stem and roots assays are similar to in vivo leaf assay. So I collected several plant materials again, and I collected macadamia leaves from clonal materials. These are from five macadamia commercial cultivars. And so here I did the implanter assay, and I did I used the method as described earlier. And so results shows that that the cultivars, that there were no significant difference in terms of stem diameter, in terms of the root biomass, there were no significant difference. So it shows that Phytophthora cinnamomi and Phytophthora multiflora are able to significantly affect cultivars 816 and 816, while there was low lesion development in cultivars 246 and 344. And so in the root assay, it shows here that we have cultivar 816 significantly impacted by post-inoculation by both Phytophthora cinnamomi and Phytophthora multiflora. And so some parameters, including the decline in fresh root biomass, were causes for us to decide how much impact it has on these cultivars. And so we have similar expression of disease showing from in cultivar 816 and cultivar 816, and the same again similar to in the stem, similar to the fresh root biomass. We have 246 and 344 showing the same. And so again, in order to also progressively confirm, I did an in vivo leaf assay. The same result, and I got similar disease expression after post-inoculation with Phytophthora cinnamomi and Phytophthora multiflora. And in terms of phenotypic ranking based on the varietal resistance, it shows that cultivar 344 was more resistant while to the diseases or caused by Phytophthora cinnamomi, while cultivar 344 again was with the most resistance again to Phytophthora multiflora. So it's the similar expression of disease based on Phytophthora cinnamomi and Phytophthora multiflora. So a conclusion here is that there is distinct partition of cultivars to Phytophthora resistance regardless of the clonal or open pollinated seed. So again, I wanted to check if it is possible that the wild Macadamia germplasm will be a good resource for a good genetic resource to identify disease resistance. And so I published this article again in Annals of Applied Biology. So I wanted to test the reaction of wild Macadamia germplasm to Phytophthora cinnamomi and Phytophthora multiflora. And I have used assertions of wild Macadamia germplasm, which are potential genetic resources for disease resistance. And again, here, there is actually limited information about if wild germplasms are actually good results against Phytophthora infections. And a question here pops up that are the same genotypes resistant to Phytophthora cinnamomi and Phytophthora multiflora? In my research, I hypothesized that there is a strong differential interaction among Macadamia genotype with Phytophthora species. And specifically, I tried to determine the resistance level of the four Macadamia species to Phytophthora cinnamomi and Phytophthora multiflora, and also check what response of these wild genotypes of each Macadamia species to cinnamomi and multiflora, and also see if the field performance of the wild genotype based on the disease symptoms, examine the performance of the wild genotype based on the Phytophthora disease symptoms. So in order to do this, I collected from an ex situ conservation site, I collected about 153 Macadamia wild genotypes, materials from them. That's leaf material anyway. And I assessed, some of the data I collected was about a tree health, the presence or the absence of stem canker, the presence of slow decline. So I developed a rating system from healthy 0 would be healthy appearance, 1 would be moderately healthy, and then mildly stressed and severely stressed plants, as shown here in the image below. So from my assessment on the general disease progression, from what I collected and from my pathogenicity trial, I saw that Macadamia ternifolia was grossly the most susceptible of them all. So here's just to show a pictorial evidence of the pathogenicity trial using macadamia leaves. So in the in vivo trial, I separated the results here, unlike before, or for Phytophthora cinnamoma and Phytophthora multiflora. And I observed that consistently, Macadamia integrifolia was susceptible to both Phytophthora cinnamoma and Phytophthora multiflora, and which has the highest lesion. Generally from this form, I subjected all the data I collected from the 152 samples, and generally I found that there were significant variation in the disease progression for each of the accessions. And so over 60% of the population were highly susceptible and with greater than 30% lesion area on the leaf samples. And here, on the far right, on the highlights in blue, we can see that a few resistant material that were consistent, those were about 15%, showing only less than 15% lesion area. Again, over 40% of the population of the wild Macadamia tetraphylla genotypes were susceptible. And that's just to give us an inkling that Macadamia tetraphylla is less susceptible to Phytophthora infections than Macadamia integrifolia. So I did the same pathogenicity test on Macadamia ternifolia and Macadamia jansenii, and it showed that Macadamia ternifolia and jansenii were really mostly moderately or less susceptible to infections caused by Phytophthora cinnamomi and Phytophthora multiflora. So within the germplasm, there were plants, there were hybrids that were also planted, and I collected those samples, and results showed that from the hybrid, there were few, at least 2 significantly less susceptible materials, which are really, really resistant to the infection caused by Phytophthora cinnamoma and Phytophthora multiflora. So here, a conclusion here that I observed from my findings, where there were about 14 Macadamia tetraphylla and 2 Macadamia integrifolia genotypes that were identified as resistant to Phytophthora cinnamoma and Phytophthora multiflora. And also there were Macadamia ternifolia is really, as shown from my experiments, that to be highly susceptible to Phytophthora cinnamoma, and only a few, I must add here that only a few Macadamia ternifolia is available as in the wild, because their population is almost going extinct, and hence the germplasm might be termed as vulnerable to infections by Phytophthora cinnamoma, general Phytophthora infections, if there is no intensification of conservation efforts in there. So currently I'm having my future work on Pythium and Phytopythium. And why am I doing this? These are also pathogens reported in other crops, but we do not know what they do in macadamias. And I'm currently examining, because during my collection of Phytophthora in macadamia, during my course of work through my PhD, I collected lots of samples of Pythium and Phytophthora, and I'm currently examining what do they do, and do they have a synergistic effect in causing root rot and stem cancers in macadamia. And it will be interesting to also know if there is a genome-wide association studies to identify markers controlling resistance in wild macadamia germplasm. And these are current work that I'm analyzing based on previous data and the data I'm collecting now. So with that, I would like to say in a nutshell that I successfully determined the identity of Phytophthora species causing diseases in macadamias, and I found new strategy to identify genetic resources in the ongoing battle against Phytophthora in macadamia in Australia. So I'd like to thank the University of Queensland for my research scholarship, and I also thank the Arts Innovation and the Macadamia Funds for providing funds to engage in this research. And I also like to thank the Macadamia pathology team, the ecosciences pathology team, the building where I did all this work, and the Maritime Research Station team, who have supported me through all my collections of the wild germplasm material that I use for my experiments. I also like to thank all my advisors who supported me through my research work during my PhD, Professor Akinsomi, Professor Bruce Top, Professor Andre Drent, and Dr. Julian Anderson. Thank you for listening. Thank you for taking time out to come for this webinar. And I've successfully been able to complete this presentation. So I would like to welcome some questions. Thank you.
Thank you, Olumide. That was insightful into the world of macadamia and Phytophthora interactions. And it's good to know that we have more than one organism we are dealing with, and also we have hope that we can find resistant germplasm to deal with these pathogens. We have a few questions here. One from Vivian regarding your isolations from asymptomatic trees. What are the requirements for Phytophthora cinnamomi to start developing symptoms in the plant? For instance, in the asymptomatic trees where you found you isolated, what are the requirements for symptom expression? When do they start showing symptoms? You are mute.
So for symptoms expression, so disease might not show for a long time until the plant is significantly stressed from lots of moisture stress, and high moisture and no moisture is a stress trigger for the plant. And once post-infection, once these weather conditions also have been fulfilled, you might begin to see symptoms as well, even from the asymptomatic trees.
So stress is a precursor to disease expression in some cases.
Yep.
Okay, that's another question from South Africa this time around. Do you have treatment for Phytophthora infection in Australia?
Yes, I'm aware that current management practice is from trunk injection, spray of metaxil, systemic fungicide, fosacid, and metaxil. But there is no, you can't, once infection has occurred, there's really nothing you can do. You can only keep managing the tree.
And the other way we've developed in our own research is soil health management.
Soil health. Yeah.
Using compost. It's very useful tool to suppress or even reduce infection. These other questions, I have one from an anonymous attendee. Does the mating type of Phytophthora have a significant morphological difference? If so, in which stage of the cycle?
So the mating types, you have to, that's why you would need a tester to know if they are different mating types. They don't present differently.
So
you have to know what the population, if it's of A1 or if it's of A2, and then once you have population explosion in terms of the production of the oospores, then you will know that for those that are heterothallic, then once you have production of oospores, then you know that there must be present two mating types in that population.
But it's difficult to differentiate them morphologically.
You really cannot.
So you need the molecular biological comparison of whether they are able to mate or not. Okay, there's another question around zoospore suspension. In your Macadamia olive assay, did you use zoospore suspension? If so, how did you induce zoospores?
So in one of the things I did was not to use zoospore suspension. I have used total colony forming units, which is the mycelia plug. So because where I've when I conducted my experiment, I noticed that once I suspend the mycelia plug, after a few hours, the production of zoospores is activated, and then they will generally respond to the stimuli of the leaf that I have dipped into as the inoculum for the disease progression.
Well, I want to say thank you. There are other questions here around control, eco-friendly treatment, which please get in touch with Olumide or myself. We can answer some of those questions. However, we've got to move on. We are running out of time. I want to thank everyone for attending the conference, but before we go on, I want to thank Olumide for taking the time out to present PhD research and also to let us know that she's looking at expanding to Pythium and Phytopythium in macadamias. For next seminar, I would like you to, if you could go to the next seminar.
Yeah, I'm trying to.
The next seminar next week is going to be a student presentation. The seminar will be focusing on 3 students who will be presenting at the Australasian Plant Breeding Conference. So they will have a sort of a trial run presentation next week. Please join us to listen to them next week. And if you have some feedback, we'd like to hear from you, or you want to have access to previous seminars or suggestions, we will be happy to hear from you. If you go to the next slide, Olumide, that would be nice. And just use these QR codes to make comments or to provide feedback. Thank you everyone for attending, and we hope to see you next week. Thank you.
All right, thank you.
Bye.
Bye.